All right. Well, thank you so much, Lisa, Heather, and the rest of the Neuroendocrine Cancer Foundation for having me on today. So my name is Rohit Thummalapalli. I'm a gastrointestinal medical oncologist at Memorial Sloan Kettering Cancer Center in New York. I specialize in the care and research for patients with neuroendocrine cancers. And today I'm really excited to talk about a recently opened clinical trial of a novel DLL3 antibody drug conjugate called ZL-1310, which I think has potential for patients with advanced, poorly differentiated gastroenteropancreatic and other neuroendocrine carcinomas. So here are the key takeaways that I'd like to discuss today. ZL-1310 is a novel antibody drug conjugate targeting the cancer protein DLL3. ZL-1310 has demonstrated a promising safety profile and early evidence of clinical activity in patients with small cell lung cancer. Gastrointestinal and pancreatic poorly differentiated neuroendocrine carcinomas, or GEP-NECs, do also frequently express the cancer protein DLL3. And so a new clinical trial testing the safety and efficacy of ZL-1310 for patients with GEP-NECs and other neuroendocrine carcinomas beyond small cell lung cancer is now open to approval, and the subject of what I'll talk about today. So before I begin talking about this new agent and this clinical trial, I wanted to briefly review our approach to management of patients with neuroendocrine cancers of gastrointestinal and pancreatic origin. This is also referred to as gastroenteropancreatic neuroendocrine neoplasms, or GEP-NENs. I know that's a mouthful. So when we think about GEP-NENs, they are broken down into our well-differentiated neuroendocrine tumors, often divided into grade 1, grade 2, and grade 3 tumors, and then our poorly differentiated neuroendocrine carcinomas, also known as PD-NECs or NECs. So in contrast to patients with the well-differentiated neuroendocrine tumors, who are often somatostatin receptor avid and are often treated with somatostatin analogs like octreotide and lanreotide, or radioligand therapies like Lutathera, or targeted therapies like everolimus or cabozantinib, patients with poorly differentiated neuroendocrine carcinomas are almost always treated with chemotherapy and really are in need of for new therapies beyond platinum-based or other conventional chemotherapies. So DLL3 is a new treatment target in patients with neuroendocrine cancers. DLL3 is a protein found on cancer cells in about 70 to 80% of patients with small cell lung cancer, type of neuroendocrine lung cancer, and neuroendocrine prostate cancer. And the best data we have so far for a DLL3 therapy, really in any solid tumor, is an immune-based therapy called tarlatamab. Tarlatamab is a DLL3 CD3 T-cell engager, which is a type of immunotherapy to treat patients with small cell lung cancer, and recently achieved early FDA approval for patients with small cell lung cancer last year, with a recent randomized phase 3 study showing benefit over chemotherapy for patients with small cell lung cancer and prior initial chemotherapy. And so full FDA approval of tarlatamab in small cell lung cancer is expected. But it's important to remember that there are a number of additional classes of treatments that target DLL3 beyond immunotherapies or T-cell engagers. And so one of these classes of drugs is a class of therapies called antibody-drug conjugates, or ADCs. ADCs are new cancer therapies consisting of antibodies which bind to certain targets on cancer cells, linked to drug payloads, often a molecule of chemotherapy. And so the rationale for an ADC is it's designed to selectively target a cancer cell instead of normal cells by selectively targeting a protein on a cancer cell. And the rationale is that an ADC may be able to deliver very high doses of the chemotherapy payload selectively to cancer cells and avoid targeting normal cells and hopefully minimizing toxicity. And so multiple ADCs are already approved in solid tumor oncology. These include Enhertu or trastuzumab deruxtecan for patients with breast cancer and other solid tumors, and Padcev or enfortumab vedotin for patients with bladder cancer. But we do not yet have an approved antibody-drug conjugate for patients with neuroendocrine cancers. So ZL-1310 is a novel antibody-drug conjugate, or ADC, developed by Zai Lab, which uses a novel DLL3 antibody conjugated to a chemotherapy payload known as a topoisomerase inhibitor, with 8 molecules of chemotherapy linked to each antibody. This is also known as a drug-to-antibody ratio, or DAR, of 8. In mouse models of small cell lung cancer, ZL-1310 showed promising activity, which has therefore led to an ongoing phase 1 clinical trial of patients with small cell lung cancer. So here is the preliminary safety data for ZL-1310 in patients with small cell lung cancer, and this is among patients that were treated as of this year's ASCO meeting. This was presented by Manish Patel at ASCO 2025. And so at the time of data cutoff in June 2025, 89 patients had been treated with ZL-1310 in small cell lung cancer to date. ZL-1310 has been shown to be well tolerated at doses below 2.0 milligrams per kilogram every 3 weeks. Among these patients, the frequency of severe or grade 3 or higher adverse events related to treatment has been approximately 6%. Most commonly, low white blood cell counts or neutropenia, low red blood cell counts or anemia. At higher doses, ZL-1310 can be associated with the more frequent incidence of low blood counts, and in about 5% of patients can lead to severe lung inflammation or pneumonitis. But in patients treated with less than 2.0 milligrams per kilogram, no cases of severe pneumonitis have been reported. And in addition, to date, among patients that have been treated at doses less than 2.0 milligrams per kilogram, only 4% of patients have developed side effects related to drug that required a dose reduction, and no patients have experienced side effects related to drug that have led to drug discontinuation. Here is the most recent efficacy data presented for ZL-1310 for patients with advanced small cell lung cancer. This was also presented at this year's ASCO meeting. So among patients with small cell lung cancer previously treated with chemotherapy, 89 patients so far had been treated across 6 different dose levels, and efficacy data was available for 74 patients. So among these 74 patients, 51% of patients were found to have major cancer shrinkage on preliminary assessment, also known as an unconfirmed partial response rate. The evaluation of duration of response, or for how long ZL-1310 remained effective, remained immature at that time. But based on this efficacy data and the previous safety data, a larger phase 3 trial of ZL-1310 at 1.6 milligram per kilogram every 3 weeks is now planned for patients with small cell lung cancer. Now, despite our progress in targeting DLL3 in small cell lung cancer, whether other neuroendocrine carcinomas that originate outside the lung, including those of gastrointestinal or pancreatic origin, can respond to DLL3 targeted therapies still remains uncertain. And so our research group at Memorial Sloan Kettering and other groups have been very interested to understand whether patients with gastrointestinal or pancreatic neuroendocrine tumors express this DLL3 marker and potentially could be candidates for DLL3 targeted therapies. And so, as you can see in the top left here, our group has demonstrated that approximately 70% of patients with poorly differentiated GEP-NETs express DLL3. Among well-differentiated neuroendocrine tumors, or NETs, DLL3 expression appears to be generally restricted to our high-grade tumors or grade 3 tumors, with approximately 40% of patients with grade 3 pancreatic neuroendocrine tumors expressing DLL3. And so the summary, the data suggests that DLL3 targeted therapy should be highly considered for patients with poorly differentiated GEP-NETs and should also potentially be considered for subsets of patients with high-grade or grade 3 pancreatic NETs. So given ours and other groups' data demonstrating frequent DLL3 expression in GEP-NETs, we've been excited to partner with Zai Lab to help design and open an ongoing clinical trial of ZL-1310 for patients with neuroendocrine carcinomas beyond small cell lung cancer. And so patients can be treated in the study in one of two cohorts. Cohort 1 is for patients with advanced gastroenteropancreatic neuroendocrine carcinomas. These are of patients who have neuroendocrine carcinomas of esophageal, gastric, pancreatic, gallbladder, hepatobiliary, colon, or rectal primaries. Patients must have demonstrated progression of their cancer or intolerance to 1 to 2 prior treatment regimens for their disease, including chemotherapy regimens. Notably, patients are allowed to receive prior DLL3 targeted therapies. These include other drugs like tarlatamab or brixtamig. And confirmation of the presence of DLL3 protein on a biopsy is not required prior to enrollment, although patients must have either a new biopsy or sufficient tissue from an old biopsy for analysis prior to participating in the study. Patients with active brain or spinal cord involvement of their cancer or prior exposure to antibody drug conjugates with the same payload, topo-1 payload, or have a history of severe lung inflammation or pneumonitis are not permitted to participate due to safety concerns. So as I mentioned, based on the safety and efficacy data from the ongoing small cell lung cancer studies to date, a treatment dose of ZL-1310 at 1.6 milligrams per kilograms every 3 weeks has been chosen for this study. And importantly, patients are treated with this ADC in the outpatient clinic, so either the clinic or the infusion center. There's no need for any hospitalization for treatments on this study. For the phase 1b portion of the study, the primary objectives are to evaluate safety and tolerability of this dose of ZL-1310 in this patient population and evaluate preliminary anti-tumor activity in the GEP-NEC cohort. And also as well in the second cohort, which is targeting patients with other neuroendocrine carcinomas. For the phase 2 portion of the study, which is focused on patients with GEP-NECs, primary objectives are to, again, evaluate anti-tumor activity, defined by the percent of patients with major tumor shrinkage or response. And secondary objectives are to further evaluate safety and tolerability. And so here's a brief statistical design of the study. So in cohort 1, the GEP-NEC cohort, we'll start by treating 10 patients. If at least one patient experiences major shrinkage of their cancer or a response, we will treat an additional 10 patients to evaluate preliminary efficacy in a total of 20 patients. And if promising, the study will expand to include 40 more patients in the phase 2 portion of the study. For cohort 2, the other NECs and DLL3 expressing solid tumor cohorts, similarly, we'll start by treating 10 patients. If at least one patient experiences major shrinkage of their cancer, we'll enroll 16 more patients, and the entire cohort of 26 will be evaluated to evaluate preliminary efficacy. If you're a patient, caregiver, or physician who treats patients with neuroendocrine cancers and are interested in the study, please reach out and express your interest. So the study is currently open at Memorial Sloan Kettering Cancer Center in New York, and is also open at multiple other cancer centers in the US as listed here. And the contact for the Zai Lab study team is also shown as below. So thank you so much for your time, and feel free to reach out with any questions.